As a molecular biologist, I've always seen DNA as this incredible recipe book for life. But what if we could use it to write completely new recipes? What if we could turn our biggest environmental disaster into something that saves lives? Well, in our lab, that's exactly what we're doing. We're taking plastic pollution, and with a little help from some amazing microscopic allies, we're transforming it into a source for new medicines. And yeah, it sounds like something straight out of sci-fi, but I promise you, it is very, very real. Just take a second and let this number really sink in. 5 billion tons. That is the staggering amount of plastic waste that is already choking our landfills and our oceans. It's an almost unimaginable number. This isn't just some environmental problem. It's a massive failure of materials science, and it calls for a completely radical new way of thinking about it. And that really gets to the heart of it. We're not just dealing with trash here. We are up against a chemical opponent that was literally designed to last forever. So, what if we just stopped trying to destroy it? What if we started looking at this endless accumulation in a different light? What if we could see it not as waste, but as a vast, untapped resource? Okay, so here's how we're going to break this whole story down. We'll start with why plastic is so unbreakable in the first place. Then we'll meet nature's master recyclers, and then walk through the two-stage solution that lets us turn this waste into medicine. The goal? To truly close the loop on our plastic problem. Right, first things first, let's get into the chemistry of why plastic, and specifically polyethylene, is so unbelievably persistent. I mean, this isn't just stubborn garbage, it's actually a marvel of molecular engineering that's, well, become a global menace. So, plastic's secret to immortality? It lies right here, the carbon-carbon bond. I want you to picture a perfectly smooth, locked chain. It's nonpolar, which means it doesn't have any of those electrical hooks or handles that water or microbes could grab onto. That bond is so stable and so strong that nature's usual cleanup crew, the enzymes and bacteria that break down things like wood or leaves, they just can't find a way in. The plastic is essentially invisible and indigestible to them. So if we can't break this chemical bond ourselves, we need an ally. A specialist, right? So we turn to the organisms that have spent literally millennia evolving to break down the toughest stuff on Earth, common soil fungi. They are nature's master recyclers. We chose a very specific fungus called Aspergillus nidulans. This thing is a biological powerhouse. It's famous for being able to produce an incredible variety of useful chemical compounds. But, and this is a big but, if you just give it a piece of a plastic bottle, it has no idea what to do with it. The plastic is just too big and chemically locked up. So we realized we had to find a way to prepare the meal for it first. And that challenge is what led us to develop our two-stage approach. The best way to think about it is as a partnership between human-led chemistry and nature's biology. We handle the heavy lifting up front, and then we pass it off to our fungal experts for the really fine chemical craftsmanship. So here's our master plan in a nutshell. In stage 1, we act as the chefs. We prepare what we call a chemical appetizer. We break the plastic down into something a little more palatable. Then, in stage 2, our engineered fungus takes over. It acts as a living bioreactor to transform that appetizer into something extraordinary. And this is how step 1 works. We take raw polyethylene waste, literally stuff we harvested from the ocean, and we use a pretty simple process with oxygen and some inexpensive metal catalysts. This whole thing basically acts like a pair of chemical scissors, just chopping up those long, unbreakable polymer chains into smaller, bite-sized molecules. And these smaller pieces are now in a form that our fungus can actually absorb and use as food. But just feeding the fungus, that's only half the story. Because what happens next is where the real breakthrough is. We're not just getting rid of plastic. We're using it to unlock genetic superpowers that have been lying dormant in this fungus's DNA for millions of years. You see, inside the fungal genome, there are these things we call silent gene clusters. Think of them like a vast hidden library of recipes for incredibly powerful molecules. Antibiotics, antivirals, you name it. But under normal conditions, up to 95% of these genetic recipes are never used. They're just silent, waiting for the right signal to be turned on. Our key to this hidden library is the gene editing tool CRISPR-Cas9. It lets us act like a precise genetic librarian. We can go right into the fungal genome, find the specific recipe book we want, and force it open. We're essentially telling the fungus, hey, don't just eat this plastic-derived food for energy. I want you to use it as the raw ingredient to make this specific molecule from this recipe right here. And this shows you the incredible difference our engineering makes. A wild fungus, it would slowly digest these plastic pieces just for basic survival. But our engineered fungus becomes this highly efficient, single-minded factory. We've essentially hijacked its metabolism, rerouting all its energy and resources to produce the exact valuable compounds we want, and absolutely nothing else.
So, what happens when we feed this pre-treated plastic to our supercharged fungus? The results are, frankly, astounding. This is the point where a global pollution problem becomes a source for life-saving medicine.
42%. That is our conversion efficiency. To put that simply, for every 100 grams of preprocessed plastic that we feed the fungus, we get 42 grams of the final valuable medicinal compounds back out. Now, this isn't just some lab curiosity. That's a commercially viable yield. And the speed of this process is just as remarkable. We get our final, purified products in just 7 days. 7. Now, compare that to the centuries, sometimes millennia, it would take for that same piece of plastic to even begin to break down in the environment. We are literally compressing geological time into a single week. And just look at what we're creating from ocean plastic. We're not making simple chemicals here. We're producing these complex molecules that have incredible value. Things like asperbenzaldehyde, which is a key building block for making other drugs, citraviridin, a vital tool for medical research, and maybe most impressively, mutaline, which is the core structure for a whole class of powerful antibiotics. We are literally turning trash into medicine. I absolutely love this quote from my colleague, Professor Burl Oakley, because it just perfectly captures the wild efficiency of this whole process. We're taking a low-value, toxic waste product and converting it with incredible speed and precision into these high-value complex molecules. It's just amazing. So let's just step back for a second and think about what this really means. This technology isn't just a clever way to clean up plastic. It's a blueprint for fundamentally changing our entire relationship with waste, for creating a truly circular future. What we're creating is a true circular bioeconomy. We're finally breaking that destructive take, make, dispose model that got us into this mess. In this new loop, pollution becomes the feedstock. Plastic waste gets pulled from the environment and is transformed into high-value pharmaceuticals. It's a system that both cleans the planet and produces essential goods. What this work really proves is a powerful new principle. By combining our most advanced genetic tools with the untapped chemistry of the natural world, we can start to solve some of our most impossible problems. And it leaves us with a really critical question. What other global challenges could we solve if we finally learn how to read all the silent recipes hidden all around us in nature's DNA?